Hello and welcome to the NGS Webinar Series hosted by the Next Generation Sequencing Competence Network. Along the series, you will be hearing from scientists and clinicians from various fields of research. However, they are all connected by sequencing. Who would have guessed, We bring you to journeys where researchers are telling us about their sequencing data harvesting and bioinformatics analysis and surely results and perspectives. How do they do it? What is it good for? Watch and let us know. Our network has started as a DFG-funded national collaborative initiative in 2019, and up to 2023, there have been several annual calls for projects to be sequenced at our core genomic facilities across Germany. From 2024 on, you have a new exciting opportunity to co-apply with one of our genomic facilities for a high-cost sequencing project, which can cost up to 1 million euros. If you ask me, it's worth to check it out. Go for it. We'll leave the link in the footnotes, and here is QR code for your fast access. Welcome aboard. Find a comfortable seat, get a drink, and let's listen to our next speaker, shall we?
All right, so good morning, good afternoon for people who are not in the SS time. My name is Joanna Perez. I'm project leader at the West German Genome Center and also NGS expert for the Competence Network of Germany. So my passion and my work focus on genomics, especially in long-read sequencing and how we can use these technologies to understand life, and in this case, how pathogens shape global health today. So imagine a village deep in the Amazon with about 6,000 inhabitants, where the river is the only road and the hospital is days away. So now imagine that in this village, there is a single clone of a deadly pathogen that is silently infecting children, parents, grandparents, with anyone realizing it. And that was the reality in an indigenous municipality in the south of Colombia that is called Puerto Nariño in 2016. And this is the story that I want to share with you today, how high-resolution genomics gave us the tools to detect, to track, and to understand this outbreak in ways that traditional methods simply couldn't. So let's start with the big picture. Tuberculosis has killed more people than any other pathogen in human history, even more than the plague of the smallpox. But tuberculosis is not just a disease of the past, but it remains the leading infection killer worldwide. By 2023, there were only 10 million cases with 1.3 million fatalities and about 500,000 of cases of multi-drug resistance. In Americas, which is the focus of this research, 3.1% are accounting for the total cases of the world. And in Colombia, especially, the weight of this disease falls proportionally on the indigenous populations. That's the same in indigenous communities in Canada, that is the United Community, or the First Nation indigenous communities in Australia. And why? Because tuberculosis doesn't spread in isolation, but it spreads in poverty, malnutrition, inequality. So tuberculosis is not just an infection, but it's a mirror of inequalities. And that's why tuberculosis is a major public global health threat, because many cases go undiagnosed. About between 2 or 3 people of 5, if they get treatment. It also hits heavily those patients that are with medical conditions, that are in malnutrition. And also there is a big gap in funding. So it's about 13 billion is required for tuberculosis healthcare, but 80% of the funds are coming from the pockets of the patients, which is not good. And there is also not funds for research enough. And unfortunately, there is still no universal coverage. So there are many parts in the world where people don't have access to tuberculosis treatment as it should be. So how does this tuberculosis disease spread? So there is a person who is infected with the disease, and this person is coughing, and in these tiny droplets there are the bacteria that is inhalated by a healthy susceptible person. The bacteria goes to the lungs, and then we have the immune cells called macrophages, and these macrophages are going to eat the bacteria, they engulf it, and then there is an immunological reaction. And what is going to happen is that a containment will be formed, like the bacteria will be a prisoner, and these structures that are called granuloma will be in the lungs. So tuberculosis is tricky because most of the people don't get sick right away. Today, 1/4 of the world population, about 2 billion people carrying latent tuberculosis. That means 1 of 4 people worldwide. So imagine this Zoom call, like if there were about 100 of us here, 25 will be already infected silently with the disease. But they don't have symptoms and they cannot transmit it. Only 5 of 10% will progress to an active disease in their lifetime, especially if the immune system is affected. And this is why makes tuberculosis so challenging, because transmission is often silent, infections accumulate over years, and outbreaks grow long before the health systems even notice. So traditional epidemiology like contact tracing, casing counts, surveys is essential, but for tuberculosis and other infection diseases is often not enough. This is where genomic surveillance comes in. So what does that mean? Tuberculosis genomic surveillance use the DNA sequencing of the tuberculosis bacteria to track how it spreads, how it changes, and how resistance is to medicine. So a tuberculosis genome is about 4 million letters long. Sequencing is like reading every single letter to see where the mistake happened. In simple words, reading the TB DNA is like checking its fingerprinter so that we can understand where it comes from and how to fight it. So by sequencing, by doing whole genome sequencing of this bacteria, we can track how the strains are spreading over the world. We can identify families of these bacteria. We also can detect drug resistance, and we can also identify patients that are in clusters and in outbreaks. So on the right, you can have like a global view of how these families are dominating the world. So on the red circles is the Euro-American, and the Euro-American is the one that is spread in all America, parts of Europe, and in Africa. And we also have other families like the Central Asia or the East African India that is mainly in Central and South Asia. And we have the West African here in green and brown that are concentrated in the West African region. So what we can see also here is that there is a host pathogen interaction that led some strains like the West African to be only focused in that region with that population, but there are others that are all over the world, and they also have some sublineages. So what is happening here is that this disease or this bacteria is fascinating, at the same time is scary, because how tuberculosis pathogen has been present in human history since the migration of the first humans out of Africa. So you can really recall in classic music and history and war philosophers, there was always a character that was infected with this bacteria. So this is the key, like genomic surveillance turns a bacteria isolate into a map of transmission. It's like moving from a blurry photograph to a high definition video. And this is now what we have, this concept of the evolutionary medicine, because we need medicine and evolutionary biology work together to understand how these outbreaks are happening and how can we treat the patients, because some of these families are really heavy. They have virulence factors like the Beijing family, so it's not the same getting infected by a Beijing strain that by a Euro-American lineage. So how we do this? So we start with the sample from the patient. So, and then we culture, then we sequence the bacteria, and then we identify variants by mapping those reads against a reference genome. These variants are also called SNPs, single nucleotide polymorphisms. Then we obtain a SNP table where we have the positions in the genome, the mutations, and based on that, we can do drug resistance prediction. We can identify the families, and we can also detect clusters of transmission. And this is the goal, that we get a report and the clinicians, they can do decisions faster. So in many countries, especially those that are high endemics, the sequencing is not there on the public health practice. So not as developed like in the UK or in the Netherlands or other parts of the world, like in Africa, in South America, or parts of Asia. This is not the situation, but this is the goal, and this is how important is sequencing technologies to study the disease. So, but it comes with challenge, especially in repetitive regions of the genome. But let's make a pause here for a second, and I'm trying to understand what is an outbreak. So an outbreak happens when a disease case appears in a number higher than expected in a community. Not all the outbreaks are the same. The normal outbreak, that one could call normal, is when it's genetically diverse, so many different families at the same party, for example. But when we have a clonal outbreak, it's one strain that is spreading person to person. It's like having identical twins everywhere. And here is the problem. Clonal outbreaks are the hardest to investigate genomically because the bacteria looks almost identical. And here is what it matters. With short read sequencing on the left and the standard reference genome, repetitive regions are poorly resolved with short reads. Variants get miscolored, the outbreak look deceptively uniform. But with long read, we can have more extension of the region, and we will include a local reference genome of the outbreak and not a laboratory strain. We can have a better picture. So having short reads is like having fragments of words, but having long reads is like having full sentences. And repetitive regions are like copy-pasted paragraphs that are very confusing for the short read technology. So after this introduction, let's get into the Colombian Amazon history. So this genomic investigation I conducted together with colleagues in Germany and in Colombia, and was published by the American Society of Microbiology in 2023. And it focused in the transmission of this bacteria in the Colombian Amazon in an indigenous population of about 6,000 inhabitants. And again, what makes a clonal outbreak different from a normal outbreak is that here most of the patients are infected with the same strain of bacteria, which makes it much harder to untangle how the disease is spreading. So the main aspects I was focused here was to bridge this knowledge gap first by identifying what was the strain that was circulating, also identifying the transmission change, and also to explore alternative whole genome sequencing approach analysis to have a better resolution of this clonal outbreak. So by 2016, me and colleagues, we were traveling to Puerto Nariño. It's in the south of Colombia, the Amazon, which has borders with the Brazil and Peruvian Amazon. So there is a remote indigenous community in this area. Those circles that you see here are indigenous settlements that are like on the right You can see some of this how a settlement look like. They are wooden houses, and the rivers are the highways. Families are living here in these crowded wooden houses, like big families of 10, 12, 11 people, and they have limited access to TV services. So we conducted one of the largest TV screening in that region, and that involved an interdisciplinary team of anthropologists, social workers, nurses, molecular biologists, the same community, because indigenous communities, they have their own language, their own politics, and their own beliefs. So many of them still believe that going to modern medicine is not the way to go. They believe in spirits, or they believe in other things, and this also delay the situation. And especially in the Amazon, where there are other infections like dengue, malaria, and many others. So then every week they have to decide, well, who is the person who is bad, really bad, or malaria, or the tuberculosis person, or all together. So, and that was, yeah, a meaningful work that we did there, but I didn't have this genomic picture at that time. So when we did this screening, we did 6,310 patients. So we screened the whole community, and we identified about 1,042 patients as systematic respiratory subjects. And from there, we identified 80 cases and could isolate 74 strains, each strain for patients. So from an outsider perspective, it could sound like, oh, it's just 74 from 1,000. But in fact, if we look this in a epidemiology or a perspective, this is about 1,236 cases per 100,000 inhabitants. And this is alarming, and this is pointing that there is a public health situation happening there. And it's like a ticking bomb that was happening at that time. So to address this, we did the classical microbiology. We have to grow the bacteria to point out this bacteria grows in 1 month. And that's why sequencing is relevant, because you can have results in a short time rather than waiting for microbiology. But in Germany, we use the genomic analysis. We did next generation sequencing, short read sequencing of all the outbreak genomes. And we implemented different approaches. So normally for the standard analysis, you use the A30RB reference genome. It's a laboratory strain, and we exclude the repetitive regions and the resistant genes from the analysis. This is the normal way to go. Then we include the same reference, but including the repetitive regions and the resistant genes. And then we generate outbreak reference genomes by choosing one of the local strains that were part of the big clonal node, and we use PacBio sequencing to do so. So here you can have again a picture of the community, and what I want you to see is that the circles that are with colors are the settlements where these patients were coming from. And when we apply the standard molecular epidemiology that is not included sequencing, what we can see clearly is that there is a clonal outbreak that was driven by a Latin American Mediterranean strain. So as I showed you before, the Euro-American lineage is all over Americas, Europe, and parts of Africa. But within that lineage, there are subfamilies that are also being selected for some regions of America. No? Latin American is mainly in Latin America strain, and we saw that this strain was across all the 16 indigenous settlements along the rivers. So the main aspect here also, we also consider the clinical data, is that half of these patients were children, and they were heavily impacted for the disease. So when we started our first approach using whole genome sequencing, here we can see that the main node was reduced, or we clearly could identify two clusters, the cluster 1 and the cluster 2. They are close, and this cluster 3 and cluster 4, they are far away. There are several SNPs of variants away from the other two, so one could say that these were independent transmission chains. So, but the transmission chains that were all focused are these ones here, and we also identify a multi-drug resistant case. So here, anyways, we can see that most of the patients carry nearly identical strains. So in this big note, there are 42 patients that are having the same strain, and here we have 18. So when we look at this, we really, I can say or think that this is deceptive because it's quite simple. But when the resolution still was limited with whole genome sequencing, so the picture was good, but it's still blurry at the edges. And when we're looking at the tracing contact data, patient data, we see that most of the patients that are in this big node, they are coming from this area. There is a gathering area there, like there is shopping, football, port. This is where, when you want to get into that community, you came by ship in a boat, and this is where you get in. So most likely the infection was introduced, and those communities are very isolated, and they are not so much exposed to other bacteria or strains, no? So the fact that the infection was heavily spread so fast and even affecting children is because I was speculating, no, that was something new for them, and that's why they were very susceptible. So here, the main finding was that the outbreak was caused for this Latin American strain, the 4.33 strain, with 2 transmission change and 1 multi-drug resistance case. So when you apply the second approach, in which we include variants in repetitive regions and resistant genes, these were highly confident variants that have good coverage and good trustability, and they were statistically significant. Then we could reduce even further the node to 27. And we could see other micro transmissions that were happening around the main cluster. So at this point, as many scientific evidence has showed that we cannot really trust the short reads for the variant calling on the repetitive regions, we then we created a reference genome using one of these strains that are part of this node. And then we use this as a reference. And then we include the repetitive regions and the resistant genes. And then here we can see that when we switch to a local read reference genome, that we pull directly from the outbreak strain, the picture changed a bit, and then we see more SNPs, finer differentiation, hidden transmission change that look like the one cluster was actually multiple micro outbreaks that were unfolding side by side, so resolution mattered. So one thing that I have to say here is that the A30RB, the laboratory strain, is part of the Euramerican lineage and part of this subfamily. So this picture would be even more interesting to see if you have an outbreak in Asia with the Beijing strain, or outbreak in West Africa, where you actually use a genome as reference that is from that lineage. So because the A30RB is from the same family, the picture looks a little bit similar. It's not similar as before, but it would be even more clear in those outbreaks. But the norm at that time was that most of the scientists were using A30RB as a reference, and this reference was made with short read data. So hopefully in the current studies or in the future, the people are using long read reference genomes, and they are also considering the diversity of the outbreak. And this is why resolution matters, and with extended analysis, this helped to increase the outbreak resolution. And then came this big surprise in our eyes, because while analyzing the outbreak, we expected to see the transmission change. Here you can see cluster 1 and cluster 2. These are the 2 transmission change. And we expected to be this transmission change genetically uniform, maybe small mutations over time, but nothing dramatic. But what we actually found was far more complex. So here in these clusters, you see col1, col2, etc. These are the patients, and on the right, on the X axis, we see the 16 SNPs, 16 variants. And what we saw is that within the patients, we saw mixed infections. So this is a heat map that shows the variants, the SNPs, and this is the frequency. And what we could see is that each row represents a patient sample and each column represents a SNP in a specific gene. The colors indicate how much common those mutations were. So the purple means low frequency and yellow means high frequency. So on the left, we only just see the phylogenetic tree with the main transmission change. And what we can see is that even or yet the SNP profiles clearly show that there are subclusters. There are subclusters with distinct patterns. So even within so what I want to say is that even within what seems to be a single clonal outbreak that is epidemiological grouped within the clusters, there is genetic diversity even within that clonality. So while contact tracing may suggest that all these individuals are infected by the same strain, the genetic evidence tells a different history. Almost 1/3 carry 2 or more clones of the outbreak strain, and that's right, about 1 in every 3 patients had more than one variant of this clone. And even we found children that were having 2 different clones at once. And some of these variants were low frequency variants associated drug resistance, which is a critical situation. This was not just a reinfection between months of years, but this was happening in real time, in parallel within the same individual. So this means that people were not just infected once, but they were getting infected with different variants. And this introduced a serious clinical problem and a public health situation because it complicates the treatment because one clone may react differently to therapy. So let's say you have a variant to rifampicin, another variant to isoniazid. So it's challenging for the clinician to give the drug that is properly, the dose that is properly, and mostly this is at children. This were between months of or 1 or 2, 3 years old children. So some communities they don't use the BCG vaccination, and yeah, adding the malnutrition and all these situations they are living. And also, this also complicates surveillance because the traditional methods they wouldn't pick on this, and this is why we can see how fast clonal outbreaks disperse in real time, and from a single source or lineage, variants can emerge almost immediately and circulate together, so creating a much more dynamic outbreak landscape that we often see. So in my perspective of what we concluded is that long-read sequencing and having local reference genomes are indeed important because it will show us hidden diversity that we have remained invisible. And this is the power of high-resolution genomics, that it doesn't just tell us the infection, like who infected whom, but it also tells us how the pathogen is evolving within and between people in real time. And this is the resume of this talk. So we can see on the left how we found this outbreak, very clonal outbreak. And then when we were applying whole genome sequencing, including the repetitive regions and then creating a local reference genome, we can see how the resolution improved. And as I said before, that would be really interesting to see in other families, in other outbreaks that are happening in other parts that are not like the Air American, that is the big one, see this with other lineages. So what we can take as a home message is that tuberculosis epidemiology in this community was driven by the clonal outbreak strain. Nearly 1 of 3 of the patients carry multiple clones, and customized variant calling pipelines where you have a good deep and where you include a local reference genome gave us a more sharper and more accurate picture. And in our perspective, this approach may be a game changer to study outbreaks worldwide when you tailor the strains, the reference genome to a given genetic background. And this also turns more interesting when you look and consider the genetic diversity of the humans, because it has been seen that these examples I told you, like why this West African bacteria is so focused in West Africa, but not in America or in other parts, because there is a genetic component from the population that is living there. So, and with this, we learned that Puerto Nariño doesn't only apply this situation to remote villages. It also applies to prisons in Europe or urban settings in Africa or migrant communities worldwide. So long reads are not just better science, but also a quality tool. And yeah, so my colleagues already show you that we are always open to support scientists to work together and implementing large studies. So we have, in my case, we have here in Düsseldorf the long read expertise, and we are happy to support you and guide you. And thank you very much for listening, and I'm open for your questions.